Hi all, welcome back. Last time out we talked about general kind of connective tissue that's associated with muscle fibers, with the fascia and that you can have deep or subcutaneous and the role that it might play sometimes with, you know, compartment syndrome and issues that you might have if you constrict that area and allow fluid to build up. But now we're going to kind of keep going with muscle, but now we're going to move underneath that layer into the fibers and talk about what does the fiber look like? What makes up a skeletal muscle fiber or a cell? Okay, so we call it a fiber because it's long and linear, typically, but that is just a special name for the cell itself. Okay, so one thing that allows for it to be long, linear, stretched out is that it's multinucleated. Okay, we talked about before that cells can't be big because of the surface to volume area ratio. Okay, that as you increase in size, the volume increases faster than the surface area. This is problematic because most cells don't make their own food, at least when it comes to humans. None of our cells make their own food. And so a lot of the energy and a lot of the communication and resources has to come from outside the cell through the surface area. Okay, and you have these proteins that are associated with that surface area, and those are the ports that allow things to come in. We'll come back and we'll talk about a special one here in a little bit, how muscles work. And that material gets in. But the problem is the volume's bigger than the surface area or increases faster than the surface area. So you have a lot of open space inside the cell and not enough room to kind of associate with that. So one way around that, and one of the problems is the control center doesn't have the capability. So one way to kind of mitigate that is to have multiple control centers. So we have multiple nuclei in a skeletal muscle fiber. So it can be long and skinny as long as you have multiple nuclei, which are the control centers of that cell. Okay? The sarcolemma is just a special name for the membrane of the muscle cell or the muscle fiber. Okay. And it gets its own name because it has its own properties. It has its own proteins associated with that membrane. Okay, it has the ability, to be. It just has muscle related proteins and pumps and things like that that you don't see as much in other cells. So we give it its own name. Sarcoplasma, same thing. Okay, the cytoplasm inside the cell, that jelly, wet jelly-like material that's typically full of proteins, has a little bit of lipids, has ribosomal RNA and mRNA floating around in it. Yeah, that and of course a lot of ions and things like that. That's the internal matrix or internal solution for a skeletal muscle fiber or cell, sarcoplasma. And then special kind of to muscle fibers is myofibrils. Okay, these are long parallel fibers. Okay, and all a fiber is a protein structure. It's just a chain of amino acids. But these ones are special because you have two different types. You have a thin one, which is associated with the protein actin, and then you have a thick one, which is associated with the protein myosin. Okay, and we'll look at how these work. But together, they're connected end to end, and we call that a sarcomere. Okay, so from one kind of the combination of actin and myosin filaments that are associated with each other makes up kind of that sarcomere. And we'll look at this here in shortly. Right? And then associated with that is going to be its own endoplasmic reticulum. Remember when we were talking about cells, we talked about endoplasmic reticulum. Okay, we talked about the rough endoplasmic reticulum, which is associated with ribosomes and the site where proteins are being made. And we talked about the smooth endoplasmic reticulum, which is where things like fats are being constructed, but also it stores calcium or other ions. Okay, in this case, the sarcoplasmic reticulum is associated with the storage of calcium. Okay? And then you have some other things like a transverse tubule or the T tubule. Okay, that's going to relay electrical impulses to the sarcoplasmic reticulum. So remember I said that these muscle fibers, they can't contract, they don't work without an impulse, without an electrical signal, without an action potential to stimulate it. And that is really going to stimulate the sarcoplasmic reticulum to release calcium. Okay, but these tubules are what's connecting that, what's sending that electrical impulse. Okay, and a triad is one tubule for every two sarcoplasmic reticulums. Okay, and we'll kind of look at this here shortly in a diagram. All right. The myofibrils consist of sarcomeres, okay, which are connected end to end, and we already kind of talked about them, and we'll come back and talk about them again. And that's really what gives that striation pattern, okay, and that's the arrangement of myofilaments in the myofibrils, okay. That's what gives those kind of lines that we see in skeletal muscle and cardiac muscle. These lines, or these sarcomeres, are the result of different colored bands that are associated with different types of filaments. So we have one band, which is a light band, which we call the I band, okay, and that has actin filaments, which are thin, okay. Then we have a dark band, which we call the A band, which is myosin filaments, which are thick, okay? And myosin tends to be, well, it is in the middle, but it overlaps the actin filaments, and that's kind of what gives you a dark band. When it occurs by itself, it's kind of lighter, but together where they overlap is the darkest region. Okay? You have an H zone, which is the center of the A band, okay, and that's going to be lighter, okay, because it's not overlapping the actin. Okay, and we'll look at this here in a photo in just a second. Okay, and then you have the Z line, sometimes called the Z disc. Okay, and that's where you have the I bands meet. Okay, so an actin filament from one sarcomere meets the other. Okay, and it creates kind of a Z or boundary. Okay, and then you have an M line, and that's where the thick filaments are anchored. Okay? And so both the Z line and M line are anchoring points. The Z line anchors the thin filaments, and the M line anchors the thick filaments. Okay? So let's look at this. All right, so here we have the entire skeletal muscle filament. Okay, the fascia kind of covers it, protects it. You have associated mitochondria that are associated with it, generating energy. We'll come back and talk a lot more about that. But if we just look at one myofibril, and we pull it out, look at it, okay, it's going to be associated with the sarcoplasmic reticulum. Okay, this is going to be storing the calcium needed for interaction between the myosin and actin filament. Okay, the actin filament are the thin filaments. Okay, the myosin filaments are the thick ones, kind of in between. Okay, where they overlap is going to be the darkest region. Okay, and then the H zone is where they're not overlapping. Okay, and also out here, part of the I band is not going to be overlapping. So the A band is the entire region of the myosin filament. The H zone is the region where it's not overlapping, and then the I band is the region of the thin filament. Okay, the Z line is where two thin filaments, right here, you can see in this picture, kind of make a zigzag because that's where they're meeting, okay? And they're offset from each other. And that's what that kind of Z or zigzag kind of structure represents. Okay? Here's another way to look at it. Okay, so this is a cross section underneath a microscope. There you can see that Z band. Okay, you can see the light I band region. You can see the darker region of the A band. Okay, but then you get to see that kind of H zone where it's light and there's no overlap. Okay. Now we'll come back and talk about how these work and why it's important to kind of see how that sarcomere looks and what the difference between a thick filament and a thin filament is. Okay, so thin and thick filament. Again, the thick filament is myosin protein. Okay, so they're going to have kind of, often we consider it like a head of a myosin protein, kind of joints out and sticks out. Okay, and it is going to bind up and find the receptor spot on an actin filament, okay, or an actin molecule, which is part of the actin or the thin filament. When that happens, it's called a cross bridge, and it needs energy to form and it needs energy to reset. Okay, and we'll come back and look at this here shortly. Okay, thin filaments, actin protein. Okay, and they're associated with a couple different proteins, troponin and tropomyosin. Okay, together, troponin and tropomyosin will link together, and they prevent actin from having an open receptor, which would mean that myosin can't bind to it. So often we need to find something that can bind to this troponin to change the shape. And one such molecule, one such ion, is calcium. So when calcium binds to this, it changes the shape, it opens the binding site on actin, and myosin and actin can form a cross bridge. We'll come back and we'll look at this in a little bit. Okay, so here you can just see what I'm talking about a little closer up. The myosin heads, so I said, you know, they kind of have a head-like structure. It's a fiber that, or a filament that kind of jets out like this with a head. This is attracted to the receptor site on the actin molecules. So these little ball-like structures have a receptor site, but right now that receptor site is being blocked by the tropomyosin kind of thread or tropomyosin protein chain. The troponin is going to bind to that, and so together they kind of solidify and they make it so those binding sites, those receptors on the actin molecule, the myosin heads can't bind with them. Okay, so we need to change the shape of this so this opens up. Okay, and that's where calcium really comes in. Okay, now we can look at it across a nice big cross section again. Okay, you can have, you know, multiple nuclei throughout, okay, to provide the communication, to provide the messenger RNA to build new proteins in case some of the proteins are broken down, send signals, that kind of thing. You can have these sarcoplasmic reticuli, which is going to provide all the calcium, and it's just going to kind of be covering that myofibril. And then you can have these other kind of transverse tubes that's going to give the electrical signal that's going to release or send the signal to the SR or the sarcoplasmic reticulum, which is going to release that calcium. Okay? Together, that and the transverse tube, we call it a triad. Okay, so you got 2 cristae, 1 tube, triad. You have mitochondria associated with this. We'll talk about it more, but mitochondria are the site of ATP. It needs energy to drive this process, so you need lots of mitochondria. Okay? And yeah, we'll come back and look at this kind of structure in a little bit. So how does a contraction work? So what does it mean to contract? Okay, so to contract means you're going to shorten the muscle fiber. Okay, and so it's going to be attached. It's going to have a pivot point, an attachment point, and you're going to shorten the fiber, and that's going to move the body part. Okay. There are lots of things associated with this, and we'll cover quite a few of them. Okay, but in a nutshell, real quickly, just look at how this works. Okay, this is pretty simplistic. Okay, so the myosin head again will bind up to the actin receptor. Okay, and when it does, it'll make the myosin head change its shape. Okay, so it makes this whole filament change its shape. Basically, it pushes, it snaps it in one direction. Okay, when it does that, okay, it loses hold on the actin receptor. Okay, and the whole actin filament slides. Okay. Then ATP can reset that myosin head so it can do it again. Grab another one and slide. Okay. And it takes ATP both to have the attachment point, which is not showing here, but also to have it trigger back. So it's kind of like a trigger. So it snaps back into place, attaches, flips, snap back into place, attaches, flips, snap back into place. But every time it takes ATP, it takes energy in order for that to do that. Okay. Now, what's exciting? What's causing this to occur? Well, you have to have some kind of electrical signal. You have to have a neuron signal. And that's where the neuromuscular junction comes in, or the NMJ. Okay? And this is a type of synapses, and we'll talk about others, but this is a type of synapses where you're going to have a motor neuron send a signal, okay? And that signal is going to release stimulating molecules. Okay? And those stimulating molecules are going to interact with the muscle fiber, and it's going to cause things like sodium and potassium to move in and out of the cell. When you do that, the action potential then can send a signal to that sarcoplasmic reticulum, which is going to then stimulate the release of calcium. Okay, so let's look at the parts of that neuromuscular junction. First, you have a motor neuron. Okay, that's the neuron that's going to control the muscle fiber. That's what we call them, motor neurons. Okay, they have a motor end plate. Okay, and this is the portion of the skeletal muscle, okay, that is going to have the receptors for the neurotransmitter. Okay, so this is the portion of the muscle that's going to be associated with the neuron. They're not touching, okay, but they're associated with each other, close enough to each other that when the neurotransmitters are released from the neuron, they can interact with the binding sites on the muscle. Okay. Now that space in between, that solution, that area in between, is called the synaptic cleft. Okay? And so you have a neuron, a muscle, and the space in between. That space is where the neurotransmitter travels. Okay, so it travels out of the neuron, across the space, and interacts with the receptors on the motor end plate. Okay. The synaptic vessel is what's holding the sacs that are holding the neurotransmitter. Okay? And so they're membrane-bound sacs that contain neurotransmitters. Okay? And neurotransmitters are the chemicals that are going to be released to deliver a message to the muscle fiber. Okay? So let's look at it. So here you have a neuron. Okay, it comes down. Here's the muscle. Okay, this is that muscular plate or the motor end plate. Okay, this is the regions associated with the nerve. This is that synaptic cleft, the space in between. Okay, these are synaptic vessels. They hold that chemical that is going to cause a response. In this case, motor neurons are going to release acetylcholine. Okay, acetylcholine is going to then react with the acetylcholine receptor on the motor plate. When it does, sodium and potassium can do their thing. They can move across this membrane. Okay, that sends a signal down into the muscle, into the endoplasmic or the sarcoplasmic reticulum to release calcium. Okay? And then the calcium can do its thing, which it'll bind to troponin. Okay? So acetylcholine, it's a neurotransmitter. It's used by skeletal fibers or muscle fibers. Okay. Again, the nerve impulse is going to release it from the synaptic vessels. We'll talk about other neurotransmitters, not just acetylcholine. There are others, okay? And we'll talk about how that works. But it's going to release it from the synaptic vessels. Okay? They're going to bind to the receptors, the acetylcholine receptors on the motor end plate, just like I stated before. Okay? That changes the permeability of the membrane. Remember, sodium and potassium ions can't move across membranes. They have to be pumped across membranes, or they have to move through channels across proteins. Okay, in this case, it's going to open the channel, open the permeability, so sodium and potassium can move. Okay, that creates an action potential. We'll talk more about these in a little bit. Okay, and that action potential will cause the sarcoplasmic reticulum to release calcium ions, okay, which leads to the muscle contraction. All right, let's look at some disorders real quick that are associated with what we just covered, okay? And then we'll dive into, well, how does calcium interact with that actin-myosin kind of interaction? How does that work? Okay. First of all, the myasthenia gravis, okay, or MG disorder, it's an autoimmune disorder, and this one is directly related to antibodies that is produced by an individual. Okay, so their white blood cells are producing antibodies that attack acetylcholine receptors on that motor plate. Okay? So those receptors that are supposed to interact with acetylcholine and change the membrane composition so sodium and potassium can move across, well, those receptors are getting destroyed. Okay? And so when that occurs, the individual will often have motor reflexes that are much slower than a normal individual or someone without MG. Okay, they also can tend to have muscle weakness and muscle fatigue, though muscles will fatigue faster because it's not going to destroy all the receptors, but it destroys lots of the receptors, and so they have less receptors, so they have less interaction with the acetylcholine, and so you have less the speed at which the membrane can move ions across, and that signal can get into the sarcoplasmic reticulum. That speed is much slower. Okay? So there are other ways at which you can deal with it. You can inhibit the acetylcholinesterase, which is going to destroy acetylcholine. Okay, so acetylcholinesterase is an enzyme that occurs in the cleft that destroys acetylcholine. So if you inhibit that, less acetylcholine is being destroyed, and so it's around more often. So the few receptors that they do have will have a high likelihood of encountering acetylcholine. You can use immunosuppressant drugs that are suppressing the production of these antibodies. Okay, but again, this could be problematic, could cause the individual's immune system to be hampered. Okay, you could administer antibodies that, excuse me, that directly compete or outcompete the antibodies that the body has made, so it inactivates these antibodies. You can do a plasma exchange, although not as common, but where you're trying to replace a lot of the white blood cells, so they're producing normal or non-MG white blood cells that aren't producing antibodies to attack acetylcholine. Okay, another one that's probably more often referred to is something like dystrophy or muscular dystrophy. It's a result of dystrophin. Dystrophin is a protein that holds muscle fibers together. And so during contraction, these muscle fibers are held together with dystrophin. If that is missing or is mutated, they don't hold together, and it's a loose kind of muscle contraction. So the rebound isn't as good, so the recovery isn't as good. The muscle itself is less tense, so you get less of a pull, and you also, a lot of times you'll get a buildup of fluid that gets in between the different fibers, and that can start to deteriorate the muscle tissue and eventually kill the muscles in that region. And then Clostridium botulinum or botulism. Okay, this is an anaerobic bacteria that is often associated, unfortunately, it's often associated with like canning of foods. So individuals who can their own foods, if you don't do it correctly, you can introduce this anaerobic bacteria, and then if you eat that, okay, that bacteria will produce a toxin, okay, and that toxin tends to mess with the receptors, the acetylcholine receptors, and doesn't allow those acetylcholine receptors to accept acetylcholine, so it doesn't allow the muscles to really fire. And it can happen throughout. It can be fairly severe in some situations. It can be treated though with an antitoxin, and you guys probably know or don't know that we use some of that toxin, small doses of it, in Botox, or we have. Okay, and that will cause those muscles that cause the wrinkles to not be able to fire. And so this is why some people with lots of Botox, they don't have the ability to really raise like their eyebrows, because those muscles in that region, the receptors are gone, and so they don't have the capability of, smiling and frowning and raising their eyebrows and that kind of stuff, because the muscle receptors are gone and those muscles can't work anymore. Yeah. Okay. Next time out, we'll talk more about that actin-myosin association and how calcium kind of regulates how actin and myosin come together. And we'll also talk about ATP and energy and its role in the entire process.